So what we're going to be talking about here today are the various steps that occur in photosynthesis. And you can talk about this and get quite complicated and talk about all the electron carriers and name them all. We're going to get into some amount of detail, but we won't get into the very, very tiny details of this. It may look complicated at first, but if you think about it, if you read about it in a couple different ways, it should start to make some sense. Okay? So when we talk about photosynthesis, there's sort of two phases to the process by which the chloroplast can make carbohydrates. And the first is the light-dependent reaction, sometimes called the light reactions. Okay? And so in the chloroplast, the light reactions occur in the grana, in the membranes of those stacks, those thylakoids. And the light reactions if you sort of just look at the overall what happens, what's the purpose, light comes in and various proteins and electron carriers work to produce ATP and NADPH. Both ATP and NADPH are molecules that can transfer energy. And these molecules, ATP and NADPH, are going to be used later on in the dark reactions. Okay, so the light reactions produce these molecules or energize these molecules so they can later be used in the dark reactions to actually make glucose or other molecules. Okay? So the way this happens, the way this works, is embedded in the membrane. So you see here we have our phospholipid bilayer. Okay, this is the membrane of a thylakoid here. And embedded within that membrane are these complexes of proteins and pigments. Okay? The complexes are called photosystems. There's photosystem 2 and there's photosystem 1. Okay? We begin at photosystem 2. Okay. Just kind of may not seem to make sense, but it was named photosystem 2 because it was discovered after photosystem 1. Okay? But the first step happens in photosystem 2. So photosystem 2 is where there is chlorophyll, okay, as a pigment. And what happens is light energy is striking these molecules. We call the light has this thing called wave-particle duality, that sometimes light seems to travel as a wave, other times like a particle. But anyway, that's sort of physics. But a photon is like one sort of particle of light. Okay? And when that particle of light strikes molecules, it can energize them. It can excite the electrons in those molecules. Okay? And so that's what happens. When light hits these molecules, okay, it excites an electron. So you remember our electron orbitals, right? You have the first orbital and then the outer orbitals. What happens sometimes when a photon strikes one of these atoms, it knocks an electron up into a higher energy level. And the energy is transferred to that electron. And then that electron that's been excited contains more energy. Some of the energy from that light has been transferred to the electron. And much of photosynthesis has to do with transporting that energized electron from one molecule to another and sort of harnessing the energy from it. Okay? So chlorophyll absorbs this light energy. It excites an electron. Okay? And that's what we're seeing here. A photon comes in. An electron from that chlorophyll molecule is excited, and it gets sort of knocked up in energy level, and then it's going to progress down what we call an electron transport chain to sort of harvest some of that energy. As that excited electron gets passed from one molecule to another, we can use that, that energy in various ways. So that energy also, down here in photosystem 2, splits water. Okay, that's where we call photolysis. And the water gets split obviously into hydrogen and oxygen. Okay? That oxygen that's released after water splits is the oxygen that goes into the atmosphere. It's the oxygen plants release. It's the oxygen that we breathe in comes from the splitting of water during this stage of photosynthesis. Okay? And so when that water molecule is split, okay, it helps to replace the electrons that are lost as that electron got excited from the chlorophyll. So those electrons, as it passes through the electron transport chain, new electrons need to be added, and they come from water. And then there's also some hydrogen that's released, and we'll talk about what that hydrogen does in a minute. Okay? So in photosystem 2, light strikes the photosystem, electrons get bumped up in energy level and passed on to the electron transport chain. Water gets split, and it gets split into 2 hydrogen ions, okay, that's down here, gets split into oxygen, which is released in the atmosphere, and electrons, which are going to replace the electrons that were lost to the electron transport chain. So that's the first step. The second step, those electrons that got bumped up in energy level get passed on to what we call the electron transport chain. Okay? And basically what this is, it's passing on these energized electrons from one molecule to another. Okay? And as they move down that electron transport chain, they're losing energy. It's kind of like if you have a water behind a dam. As you let that water go from a higher energy state, flow through that dam, you can harvest some of the energy and use it to do work, to generate electricity or run a mill or something. It's sort of the same thing. We bump up an electron, and then as it comes back down to a lower energy level, we harness the energy. And in the electron transport chain, what's happening is these electrons, as they move down, the energy is used to pump hydrogen ions, okay, from the stroma, the fluid around, into the thylakoid, okay, into those discs. And so as hydrogen gets pumped in, we're building up hydrogen inside of this thylakoid, okay? And the concentration is building up and getting greater and greater. So that's the electron transport chain. And then the last step here, okay, last steps, photosystem 1, the next one, and it acts in a similar way. Light hits photosystem 1. Electrons get re-energized. And those electrons, after they get re-energized, they transform NADP, okay, into NADPH, the reduced form of that, which can then carry that energy to the dark reactions. So if you follow the path of the electrons, light hits photosystem 2. Electron gets bumped up. It goes through the electron transport chain, pumping hydrogen ions. Then gets to photosystem 1. Light hits it again, bumps the energy level up again, and then that electron is transferred into NADP, okay, to form NADPH. Along the way, hydrogen has been building up. Hydrogen ions have been building up inside of the thylakoid. And so there becomes a concentration gradient that you've been pumping hydrogen inside of the membrane from the outside. And so we know that because of diffusion, those molecules want to pass through, and they do eventually. Okay? And when they pass through, they go through the special protein. Okay? You see here, the hydrogen ions have been built up here. And for them to go through, they go through this protein, and this shape, and it sort of spins as they go through, and this protein is called ATP synthase. It basically uses the energy from this concentration gradient And as the hydrogen ions are diffusing or going through this protein, they kind of spin this ATP synthase, and what it does is it forms ATP. Okay, that's how we get the ATP from the light reactions. So, sounds like a lot, right? But it's really not all that complicated when you break it down to the basics. Light hits photosystem 2, bumps up electrons, which gets past the electron transport chain. As it moves down the chain, pumps hydrogen ions, okay? Also splits water, gets to photosystem 1, light hits it again, bumps the energy level up, and then it's passed on to NADPH. And those are the light reactions. So we know, and we've talked about this before, ATP. And what is it? Well, it's an adenosine triphosphate. It's an energy carrier molecule. It's very important for the cells. And as always, the energy is stored in the bonds, in these high energy phosphate bonds. If we look here at this ATP molecule, okay, we can see it's triphosphate. Adenosine is here. Triphosphate, you say 3 phosphate groups on ATP. And these bonds between phosphate and oxygen contain a lot of energy. And so when they're broken, they can release a lot of energy. Okay? And so ATP, when the energy is used, gets converted, a phosphate group gets broken off, and then it forms ADP, adenosine diphosphate. And again, you see here, there's just 2 phosphate groups. That can get, another phosphate can be added to it to make it back into ATP. And so you have this cycle of ADP to ATP storing and releasing energy. Okay? That ADP gets recharged by mitochondria. They add that phosphate group to form new ATP. All right. Sorry. Yeah. So all that requires light. Each step requires light hitting photosystem 2, photosystem 1 to energize the electrons, to produce ATP, to produce NADPH. Okay. So when there's no light, that doesn't happen. That stops. All right. And those products that were created in the light reactions, okay, ATP and NADPH, are needed for the next step, which is the light-independent reactions, sometimes called the dark reactions. So the light-independent reactions, also called Kelvin cycle, okay, named after a scientist that sort of started the research on discovering what's happening here. This is where carbon is fixed. So where carbon, because if you notice, and what we talked about, the light-dependent reactions, carbon dioxide, we'll take a break after we finish this section, that carbon dioxide wasn't a part of the reaction yet. Right? We needed light, we needed water, but carbon dioxide has not come into play yet. Carbon dioxide comes into play during the Kelvin cycle, during the light-independent reaction. Okay. Now, it's a little bit of a misnomer to call them the dark reactions because they don't really happen in the dark. Why not? Why don't these take place in the dark? Because they don't require light.
The other part needs light.
And make the next step. Nate?
It needs the products that the light reaction.
Yeah, it needs the products from the light reaction. It needs ATP, it needs NADPH. So if that can't be created in the light reactions, then the light independent reactions cannot go on. So it stops in the night as well. I guess I set it up there.
I totally know it.
Yeah, all right. So the light-independent reactions, the Kelvin cycle, this takes place outside of those thylakoid membranes, in the stroma, in that fluid that fills up the chloroplast, okay? And basically, it can be split into 3 phases. Okay? You have carbon fixation, when carbon dioxide is sort of grabbed from the atmosphere and turned into other forms. Okay? Then it's reduced into a form which can eventually become glucose. And then you need to regenerate the starting products that allow this cycle to repeat over and over again. So those are sort of the steps. And you know, there's a lot of different molecules that help to reduce these, and there's different enzymes. We're not going to get into too many of the details. But what happens in the light-independent reactions, first, carbon dioxide's taken in. And overall, the goal of the light-independent reactions is to produce the precursors to glucose, molecules that can eventually become glucose. So, sort of the steps as follows is carbon dioxide molecule combines with a 5-carbon sugar called ribulose biphosphate, RUBB. Okay? And that forms, obviously, we have a carbon from the carbon dioxide, we have a 5-carbon sugar, and that forms a 6-carbon sugar. But it's an unstable 6-carbon sugar that basically immediately breaks down, okay, into 2 3-carbon sugars, okay, called phosphoglycerate, PGA. ATP is used, NADPH are used to transform this molecule, PGA, phosphoglycerate, to transform it into other molecules called G3P, glyceraldehyde 3-phosphate. Okay? So they transfer. That can then be used to form glucose later on. It can be used to form amino acids or starches or other molecules. This is the purpose of the dark reactions, to produce this molecule, okay? This G3P, which can be used to form glucose. And then the remaining carbohydrate molecules need to regenerate ribulose biphosphate so that it can start the cycle over again, combine with carbon dioxide and repeat this whole thing. Okay, so you need that cycle. So again, what we have is our 5-carbon sugar, ribulose biphosphate, combined with carbon dioxide. There's a very important enzyme called rubisco that actually facilitates this. And then it forms the 6-carbon sugar that's unstable, splits into 2 3-carbon sugars. Okay, that's the PGA, phosphoglycerate. Okay, those phosphoglycerate, again, we created 2 of them. ATP is used, NADPH is used. Where did these come from? Came from the light reactor. Okay, okay, and that transforms the 3-PGA into G3P plus ADP and NADP, and eventually those molecules can form glucose. Okay? And again, they're recombined to reformulate this ribulose biphosphate to start the cycle over again. Yes?
Were those pictures like the same as the ones from the other slide? Like,
Yeah, because I separated them out. Yep, those
Oh,
same
okay.
picture. Yeah. All right. So this G3P, okay, also called PGAL. Okay, this is an important. It's a generic product that can be transformed into many, many molecules. You know, when we talk about photosynthesis, we say photosynthesis produces glucose. Okay, and it can, but actually the end product of photosynthesis is actually this 3-carbon sugar, okay, G3P. And it can be, most of it is turned into glucose, but it can be synthesized into proteins, into lipids, okay? It can be used to form cellulose and starch. So it's used for a variety of purposes, but it's a general building block carbohydrate that can be used, okay? So you can see that G3P can be transformed into other types of compounds for the plant. As we learned about in our workbook, there are various factors that affect how quickly photosynthesis can take place. okay? And obviously, the amount of the reactants is important. The carbon dioxide level, the water levels, minerals from the soil are important. Temperature is important. All these things can affect the rate of photosynthesis. And you can see here the rate of photosynthesis as light intensity increases. You could see the rate of photosynthesis as temperature increases. Now, what happens over here?
Stops when it gets
Why?
denatured.
Yeah, because as we know, these are all enzyme-mediated reactions. It requires enzymes for all these little steps to happen. And when enzymes become heated to a level, they can break down, they can denature and not work anymore. So that's why this graph looks like it's that shape.
What is like 30 degrees Celsius and Fahrenheit?
What's that? That's like 80. 37 is human body temperature.
What's the radiance?
Intensity of light. So this is the intensity of light and what happens in photosynthesis. All right, any questions about the process of photosynthesis? Okay. You guys can take a break for a little bit. Take a few minutes.